am I? I always think it's good to have a nice sort of background information on who you are. And so, as was pointed out, I did a Bachelor of Science with Honours at the University of Melbourne and a PhD in medicinal chemistry. So both of these projects were looking for phosphodiesterase inhibitors. The first one was looking for PDE3 inhibitors, and my PhD was really a traditional medicinal chemistry project where I designed the compounds, synthesized them, and then did some very basic, very basic cell-based assays under Phil Thompson. Whilst I was completing my PhD, I also decided to do a graduate certificate in commercializing science. And this is something that I'm really passionate about, and that's, you know, merging academia and the commercial world. And it's something that, you know, I try to instill in my group. As was said, I did a bit of a postdoc in some of the small, so we have a consortium of academics that make these CTXs. And so the compounds that I helped design on there reached phase 2, which was quite nice. I also did some work at Biota before joining Michael Parker's lab at St Vincent's Institute. So Michael is a crystallographer, and as was said, I spent 10 years in a crystallography lab, but as much as they tried, I actually never ever crystallized a protein. I was hired purely to do drug discovery, drug design, but also look at structure function of proteins. As I said, I was made a team leader in 2019 before being recruited to RMIT under the Vice Chancellor's Fellowship Scheme. So just a few things. I think science isn't just about what we do, it's about how we sort of give back to the scientific community. And so there's a few other things that we do a lot in the lab. The first is that I was a BSc Inspiring Woman. So this is a group in Australia that are there to encourage women in science and particularly encourage those to go from early career all the way to sort of a senior researcher. And I also do a lot of commercial contracting work, so with quite a few companies both in Australia and also overseas. So my lab is about 50% funded by these commercial contracts. Another thing I'm really passionate about is teaching science to kids. And so I'm what's called a SciRO-STEM professional, where I go into the local primary schools once a term and teach them about science. So I just want to give you a little bit of an intro into RMIT. Some people might not know that, but RMIT is a global university in technology, design and enterprise. So as you can probably see from some of these rather funky buildings, that they really are one of the world leaders in art and design, but also probably one of the world leaders in engineering and really building a reputation in science as well. They are actually a technical college, is where it started. So they have very strong links with industry, which was something that really drew me to RMIT. So we're in Melbourne. So Melbourne, for those that don't know, here's Australia. Melbourne's right down the bottom of the mainland. So we do have Tasmania below us. Most of you probably know Sydney, where the Sydney Opera House is, and the Sydney Harbour Bridge, and that's about a 11-hour drive on a nice day from Melbourne to Sydney. Australia is obviously known for its beautiful beaches, but we've also got some beautiful land around us as well. So this is actually all within about 20 minutes of where I currently live. So this creek here is down the end of my street. About 20 minutes we have some sort of semi-temperate rainforests, and of course about half an hour from where I am is the beautiful Yarra Valley, which has some of the most gorgeous wines in the world. I could spend the next sort of 45 minutes talking about where I am and who I am, but that's not what you guys are here for. You're here to talk about this protein, dynamin-1-like protein, or DRP1. So I will go into the mechanism a little bit, but I just want to start this by saying, in a nutshell, DRP1 is a GTPase that regulates mitochondrial fission. So the way it does this has really been elucidated by a lot of cryo-EM, and there's a few different mechanisms that have come to light, but the one I'm going to focus on was one that was published in 2017. where you have DRP1 with a recruitment factor, and what it does is it forms these beautiful oligomerized rings. So if we swap over to the sort of the schematic, it's a bit easier to see that these monomers recruit things like MID that allow it to form, to bind to the mitochondrial outer membrane, form these oligomers that then tighten around that membrane. A fission factor comes in, and those mitochondria are pulled apart, forming these small fragmented mitochondria. Structurally, we know that DRP1 contains a GTPase domain, which is shown in green, and that GTPase domain also contains two bundle signaling elements. It also has a middle domain, or sometimes called a stalk, which is highlighted in cyan here, a variable domain, which has not been crystallized to date, but it's sort of highlighted by these two orange spheres at the bottom here. We also have a GTPase effector domain, which is in magenta, and that's part of the stalk and also one of the bundling signal elements. One thing to note is that there's actually 9 DRP1 isoforms, and this is a review that we published this year, where we actually gathered as much information as we could about these 9 isoforms. There's really not a lot of literature out there where people have discussed the different isoforms and been really careful with their studies in saying which isoform they used. But as you can see, the isoforms really differ by an A insert, which is in the GTPase domain. So you know, about half don't contain this. And the other reason the other area that they really differ is in this variable domain, where they could have a full variable domain, not have the variable domain, such as isoform 3, or have some sort of portion of a variable domain. So these 9 isoforms are very, very different, and I've got this table here, not so much for you all to read, but just to sort of highlight a few things to you that our research in this, and this was really led by one of the PhD students in my lab, Aisha, where we really only found information about 4 different isoforms. So there were really only 4 isoforms that had been well described or distinguished in the literature. But what you can see is that they do differ significantly. So their self-assembly differs either with or without lysosomes. Also, and probably more importantly from a drug discovery aspect, is that the GTPase activity actually differs quite significantly between those isoforms, either without lysosomes, with lysosomes, or with or without these recruiting factors such as MIF. Regardless of the isoform, the role of DRP1 is pretty well established in mitochondrial fragmentation, as I mentioned. So mitochondria live in a very delicate balance between fusion, which is this sort of elongation of mitochondria, and fission, which is this mitochondria being quite fragmented. And there are proteins involved in both. DRP1 is thought to be really the major protein involved in this fission reaction. And increases in DRP1 and increases of fission and this mitochondrial fragmentation lead to somatic cell death, increased cell division, proliferation and invasion, and increased pluripotency. And so with all of these sort of outcomes, you can probably imagine that DRP1 has been implicated in numerous diseases, and probably the 3 most strongly indicated ones is cancer, neurodegenerative disease, and heart disease. So in terms of neurodegenerative disease, it's thought to be involved in numerous types. So Alzheimer's, multiple sclerosis, and Parkinson's are some examples here. But if you search through the literature, there's many examples. And there's also many mechanisms for how it has a role in here. But this one I kind of like, where if you can imagine DRP1 oligomerizing and causing this mitochondrial fragmentation. If this was to occur in a neuron, that neuron would die, leading to Alzheimer's disease, is my very simplistic summary of these. But there's many publications out about a really strong role in neurological diseases. Probably the area that is of most interest to the team that's working on our particular program is myocardial infarction, and that's because Max Lim, one of the researchers in here, has a very, very strong background in myocardial infarction. So obviously myocardial infarction is also known as a heart disease, where simplistically it occurs when blood stops to a part of the heart. And so that can cause injury in itself, but a lot of the injury has actually occurred when it's reperfused again with blood, and this is called ischemic reperfusion injury. And Max has spent a lot of time actually, and other researchers, really showing that mitochondrial dysfunction is a main feature of this injury. In particular, if we have a look at these slides here, where you have ischemic reperfusion injury, where you put this into a mouse model, you can see by these black circles here, this really fragmented mitochondria. Whereas in our control with no injury, you get these really nice elongated mitochondria. And so you can mimic this as well. So if you take MEF cells, so that's mouse fibroblast cells, which have a wild type version of Drp1, you create this injury in these cells, and you get this sort of fragmentation of the mitochondria. However, when you knock out Drp1 in these cells, you end up with elongation of that mitochondria, similar to the control. And cancer is one of the more interesting areas. So I've been funded a lot in the past by cancer and spent quite a bit of my career in cancer, and thought that it would be a really nice disease target for Drp1. However, there's quite a lot of conflicting evidence in cancer. So some of the studies have really suggested that increased levels of Drp1 are beneficial because they're associated with apoptotic cell death. But a whole bunch of other studies have really shown that Drp1 inhibition alone, or in combination with different chemotherapies, leads to much more favourable outcomes, both in vitro and in vivo, and this is across the range, quite a range of cancers. And a lot of this ambiguity really, I think, has to do with potentially the isoforms that they're using in Drp1, but also the way that they're inhibiting Drp1. So to date, there's one known small molecule of Drp1, which is called MDIVI. So this was found back in, or published, the finding of this back in 2008, and it was found from a screen that was done on yeast Drp1. If you can have a look down here of the species, you can see yeast is right down the bottom with only about 50% identity to the human Drp1. So, you know, that in itself brings up a few little question marks. Further studies on this, though, have really highlighted that potentially it's not a specific human Drp1 inhibitor. In yeast, it's got an IC50 of about 10 micromolar, 1 to 10 micromolar, depending on which paper you read. However, in human, studies in human have shown that this is more in the millimolar range. Our studies have also shown that it really has sort of modest protection in human cardiac resistant stem cells compared to those sort of knockout environments. Again, studies by Max and his team have shown that there's really inconsistent cardioprotection in animal models. And recently, the mechanisms for this are coming to light, where MDV1 interacts with complex 1 and potassium channels. So it really does seem to be quite a non-selective inhibitor. Therefore, there's obviously a need to be able to find, you know, a selective inhibitor for pharmacological reasons, as well as for a drug discovery aspect. So the method that we use in the lab, and I saw, you know, over half of you are computational chemists, so you probably have seen this method a billion times before, but I'll just quickly highlight what virtual screening is. So you take a protein structure of interest, so that could be a crystal structure, it could be a homology model, NMR structure. And you have libraries of millions of compounds. We tend to use commercially available compounds only, not sort of, you know, real space kind of compounds. You do a bunch of parallel docking calculations and analyze these computationally. And so some of the ways that we analyze these is not just looking at the score, but we obviously cluster these apart. So we're looking for diversity in the compounds that we purchase. You wouldn't want to have one compound, you wouldn't want to purchase 100 compounds and have them all look the same and all be inactive. So for this project, the screening strategy was, as I said, it was 5 years ago, was to look at the PDB at the moment for H1B was available. And obviously it doesn't have that middle domain and stalk domain, but we decided to screen in the GTPase domain. So we conducted a virtual screen. At the time, I had 16.5 million compounds from 18 suppliers that we then filter for drug-like properties and end up with about 5.3 million compounds. From this screen, we purchased 50 compounds, academic project, and went straight into cell-based assays because that was really the only assay that we had up at the time. And so what you're seeing down the bottom here is the 8 hits that came out of this screen. And so the way that we described a hit was this is MDivi, and we were looking for anything that had either greater cell death than MDivi or greater fused mitochondria than MDivi. And so we ended up with these 8 compounds that sort of seemed to be having this effect. But a cell-based assay isn't going to cut it in drug discovery, so we decided to develop an SPR assay for DRP1. So for those that don't know what surface plasmon resonance or SPR is, it's where you take a gold chip, you tether your protein to the chip, and then pass your compounds across the top of the chip. And if they're binding to the protein, they change the diffraction angle of the light, which can be measured. And so when we took those 8 compounds and screened them via SPR, we actually found 3 compounds that had nice dose response binding to DRP1. You know, not brilliant KDs, but these are hit compounds. But one thing you might note here is we also looked at MDivi against this human DRP1 and found, you know, 0 binding up to, you know, 80 odd micromolar. So we had the direct binding. We really wanted to then go one step further and see if we could get some sort of in vitro functional effect. And so we developed a GTPase assay. Both we purchased the kits, but we also developed a mass spec version of this assay as well. And so, you know, pretty simply, the sort of way that these assays work is that DRP1 naturally binds GTP, which gets phosphorylated and becomes active. If an inhibitor was to stop this from occurring, GTP then loses one of its phosphate atoms, which interacts with the green dye, and you can get this assessment of GTPase assay. And so our 3 compounds, and you know, we're really not near the KDs because they become quite insoluble as we get higher. But we were sort of seeing a dose response in this GTPase assay, which was really nice to see. So taking a little bit further, I sort of said that we'd done this initial screen against fibroblasts, but we really wanted to make sure that it was a dose response effect and also make sure that it was a selective effect in the cells. So we took the 3 of them and looked at fused mitochondria. So what you're seeing here is the greater fused mitochondria, which means that fission is being stopped or DRP1 is being inhibited. And what you can see in the wild type that all 3 compounds had a dose response effect. And you know, once again, I want you to sort of remember that this compound had a calculated KD at about 200 micromolar. So we're really nowhere near where we'd be expecting to see that effect, you know, a strong effect there. But excitingly for us, when we knocked out DRP1 and the same thing, fused mitochondria, we saw really no effect on that fused mitochondria at all. So this is really, you know, giving us that evidence that these compounds are are causing this stopping of fission due to an interaction with Drp1. So this was about the point where I had met Franca at a conference in Barcelona. And the way that we would usually go from here is trying to improve our compounds using SAR via catalogue. And so once again, I know you're a lot of computational chemists that really understand this, but I will explain that the really simple thing is you take a compound, and because we've gotten diverse compounds, we always feed this back into our compound database, and really you're just looking for compounds with different decoration on them to try and build out your SAR. And so traditionally we'll use programs like Unity to do this sort of work, but Franca was talking about the BioSolveIT challenge, and I applied, and so I had the BioSolveIT software that I could use. So I decided to use F-trees, and so this is going to be my very bad example of this, and I'm sure if there's questions, the actual scientists at BioSolveIT can answer it later on. But the way that I sort of describe F-trees is it's almost a bit like a fast pharmacophore screening. And so it takes your compound and it sort of breaks it up into these nodes, and then you can screen your compound against these nodes. So as I said, it's very, very similar to what we traditionally use as pharmacophore modeling. However, you can do it with a single compound, and it's super duper fast, much, much faster than any pharmacophore screening I've ever done before. So we decided to use both methods, and screened our 3 hit compounds against with our databases, our compound databases, so that's 6.5 million drug-like compounds, using both Unity and F-trees. So the other program that I had on Access was SeeSAR. So we redocked all of these in, and SeeSAR uses FlexX, and then ranked these compounds. So we ended up purchasing 64 compounds. These aren't it there, these are just a sort of visual placement. But I can tell you that 27 of these were inhibitor 1, 7 were inhibitor 2, and 30 were from inhibitor 3. So all of that backtracking on assays meant that we had a really nice drug discovery pipeline for these 64 compounds. And so we started with our Biophysical, obviously. And so 5 of these 64 came out as completely insoluble. When we ran all of the compounds against our blank chip, 13 of these actually bound to the blank SPR chip, so were taken out of analysis. We then ran a single dose at 100 micromolar, and 17 of these compounds showed no binding at all at 100 micromolar. 19 showed binding, and 15 showed a beautiful dose response affinity. And so I've been a bit cheeky, and I'm using an adjusted hit rate, taking into consideration the compounds, the 18 compounds that couldn't be assessed. And we've got a hit rate of approximately 53%. So one thing that was really important to note is that 12 of these were actually Drp1/3 analogues, which was really nice. Now I'd love to show you some beautiful dose response SPR here. However, in Melbourne we have been in lockdown for COVID since March and unable to enter any labs. So I have been, I couldn't go into lab and get the data. I've showed you what this is data for the Drp1 project, but obviously not the nice dose response data. In the meantime, we've also set up some crystallography in the lab because obviously, you know, we'd like to see how our hits are binding to the protein. And I can tell you that we have really beautiful diffracting 2.6 angstrom crystals that have been co-crystallized with GDP that you can see in the site there. We have done some soaks with our compounds, and at the moment they're not looking so promising, but we're doing a lot of work in trying different isoforms to see if this will improve the crystallization. And so the aim of this obviously is to do hit to lead drug design, and once again, don't really need to describe to this audience what we aim to do for hit to lead drug design, but I will anyway. So what we do is we have a compound of interest, and my job traditionally has always been to look for areas we can fill. For example, the big hole at the back. Ask the chemist to make a compound and hopefully see a jump in activity. So in this example, a 10-fold jump in activity. And obviously, it's not just about trying to find holes that you can fill or hydrogen bonds that you can make, but it's also about sort of focusing on phys-chem properties as well. So in this case, it needed a little bit of solubilizing out the front here, and we ended up with another 10-fold improvement in activity. And so in you know, an ultimate drug discovery program where you have comp chemistry involved, you know, you could essentially make 2 compounds with a 100-fold improvement. Anyone who's ever worked in med chem and comp chem know that there's 50 compounds either side that have broken the models along the way. Obviously, I can't show you the 3 compounds that are our hit compounds, but what I can say is that we do have really nice binding modes of our compounds. So we've docked the compounds using the FlexX within SeeSAR, obviously, but also using GOLD. I always like to use at least one other docking program, and managed to find a really nice consensus binding mode. So not just in the programs themselves, but also across the programs. So quite a lot of confidence in that binding mode. And so we decided to have a go at not just doing the traditional way of sort of you know, looking and seeing what we could interact with, but also using SeeSAR. And you can see with some of these sort of jelly beanie type shapes that there's at least a little bit of space that we can fill. So for those that haven't seen SeeSAR, I didn't see SeeSAR until I went to this conference a year ago. And this is an example. So what I shouldn't say I've done, what my student did was actually redock in GNP into the crystal structure, just so that I could give you a bit of visualization on how SeeSAR works. And I will say that my students in particular absolutely love SeeSAR because it's a really easy way to visualize these interactions. And so what you can get from there is, you know, green circles is good, and the red circles is work that needs to be improved. But you can very quickly have a look exactly why and how you can improve that. So in this case, you know, it's only 2.6 kilojoules per mole, and there's a nice little interaction occurring. So, you know, maybe you might want to keep that. Whereas in the case of 06 over here, you can see that it's a 15.4 kilojoule per mole hit, and there's no sort of interaction that's taking place, and it's something that you can think about changing. The other thing that, you know, and not just my students, I've got to say, but some of the chemists that I've worked with as well, really like about SeeSAR is that you can literally at a click of a button change the atoms really quickly and design new molecules that fit that space and do that calculation. And it's really quick. It's much quicker than sort of a docking experiment would be. So, you know, I would have loved to have shown you what we've done. So we have designed a whole bunch of analogues in here. But as I sort of mentioned, COVID has meant that our labs have been shut for the whole year. So we have a long list of compounds to synthesize, and at the moment, only 2 of those have been synthesized and not yet tested. But hopefully I can sort of show you that there's a really strong future for this program, in that we've used our in silico modeling to get a bunch of structural analogues and currently undergoing some medicinal chemistry, which is feeding into the in vitro assays that we've got developed, and these are quite robust assays. So the Biophysical SPR assay has been really robust, and that GTPase assay is great, and I think it will be even better when we get more potent compounds. From here, we feed these into our cellular assays, and we have quite a suite of these, mostly looking at mitochondrial activity and morphology, but also looking at survival, and also obviously we now have the knockout cells as well to sort of do that sort of cellular check of selectivity. Crystallography is always going to be an aim for this project, but anyone who's worked in crystallography knows that sometimes it doesn't work the way that we want it to. So we do have some nice docking models of this, and we do have some site-directed mutagenesis you know, thought about to kind of validate that binding site. And obviously, all of us know in drug discovery that this is a really circular process that will continue to go until we get a nice potent compound that we can, run those really formal selectivity assays on. But obviously the benefit of this program is that we've started our screening on human Drp1. MDV was started on yeast Drp1. So I'm just going to sort of acknowledge the people that have worked on this project. So a lot of this has been done at my previous work, St Vincent's Institute, and really headed up by Max Lim, who met me at a student poster night actually, and said, "Oh, I like your poster, you could help me on a project." And from there we've spent 5 years working together and trying to run this program on literally zero funding. So Aisha is a brilliant PhD student who's worked on this project and has really driven a lot of the work that's been done so far. Christopher Langendorf has been doing the crystallography on this project and has been really working on looking at different isoforms and looking at different dynamins as well. John O'Call, John Scott, Will Smiles and Naomi have done a lot of the protein expression and also developing that GTPase assay. So the chemistry is occurring when they're allowed back in the lab at La Trobe University, which is also in Melbourne, headed up by Belinda Abbott and one of her students, Jared. And Leah Delbridge at the University of Melbourne has done a lot in terms of assisting with cell culture and so forth. So I'm just going to sort of finish up by saying that the aim of my work as a computational modeler is to generate ideas. It's never to give a single answer and to say that this is, going to be your potent selective compound. It's to work with chemists, to work with biologists and generate ideas. And one of the things that I found by using the BioSolveIT software is that we can generate these ideas really quickly. Which kind of leads me to the really obvious thing of why we use computers for drug discovery. And everyone here has no doubt seen this, probably this exact picture. But the aim of using computers for drug discovery is really to reduce this 10,000 compounds to a much smaller number, to reduce this amount of probably really €1.5 billion at the moment, and reduce this time to be able to really speed up drug discovery. And if we can do that in a fast and cheap way, we're all going to be winning.